Thank you, Bernard, and welcome everyone. I would just like to say up front that I am presenting this work on behalf of all of the members of the TPCP. So in various slides, you will see some selected photos of individuals who are either driving the research or are first authors on the publications that I am showing. But just to let you know that there is a huge team behind the research that I am presenting this morning. So, taking on from this morning's discussions, and wherever you might be joining us from across the world, today we are all pretty familiar with this figure showing the global distribution of the COVID-19 pandemic from its first epicenter in Wuhan, and how in just a matter of 5 months the disease has spread, and so widely. But the situation we find ourselves in lockdown today does present me with an excellent opportunity to relate the work that has been done with one invisible pathogen to work that we have been doing in the TPCP. Where last year alone we were working with over 31 different groups of pests and pathogens, mainly affecting plantation-grown eucalyptus and pine, and I will touch on some of these examples during the talk. But just to give you some background for the talk, when we are dealing with a new disease outbreak, the first response is containment, to try and limit the spread. If the disease outbreak is small enough, it could be eradicated. Secondly, we need to identify what is causing the epidemic. Is it a virus, bacteria, fungi, or an insect? We really do need to know what we are working with. So accurate identification is the single most important part of research that needs to first take place, as it lays the foundation for downstream applications. For example, once you have a proper identification, then you can develop diagnostic tests, and this is something that Almuth will cover in her talk. Thirdly, we would want to look at how widely distributed the pathogen is, say if eradication was not successful, and this relates to the map that I showed you earlier. We also want to look at the genetics of the pathogen and the population as a whole, see if it is clonal or diverse, or if it is changing or mutating. It all becomes very relevant if we are looking at the invasive potential of the pathogen. And the last research theme relates to gaining a better understanding of the biology and ecology of the pathogen. All of this research taken together will help us develop integrated pest and pathogen management strategies or control, and this is something that Brett will touch on in his talk. But for the rest of my talk, I will highlight some examples of our work that relates to these 4 themes, not to go into any particular detail as time doesn't really allow for this, but rather to give you some food for thought for the panel discussion that will happen afterwards. So the first theme I will touch on is containment, bearing in mind that it is much easier to control and eradicate a disease in a small area where the outbreak first occurs than if it becomes widespread. And the example I have chosen to illustrate this is Fusarium circinatum. This pathogen is very well known to this audience and causes root and collar disease in the nursery, as you can see on the left-hand side, and pitch canker disease in the plantations, as you can see on the right-hand side. This is an old pathogen for the TPCP, and we have been working with it for at least 30 years. The first outbreak of the disease occurred in a single nursery in 1990-1991. Unfortunately, at this stage, the disease was not eradicated. Instead, it spread to all the nurseries across the country. It was contained for a while in the nurseries, at least for 25 years, but then it later moved on to the plantations, and there are direct links to show that it came from the nurseries. But now, what is quite alarming is that there has been a host jump of the pathogen from the plantations onto different native grass and herbaceous plant species, and this is this understory vegetation you see on the right-hand side. It is not causing disease here, so they are asymptomatic carriers of the pathogen, just like some humans are for the novel coronavirus. These plants now essentially act as reservoirs of inoculum. What this example demonstrates is that containment and/or eradication is no longer an option for Fusarium circinatum, but that instead it now can only be managed. Moving on to our second theme, where I have mentioned how important correct identification is, as we need to know this in order to implement proper control strategies. For example, it was only when researchers discovered this was a novel coronavirus, very different to the common influenza virus, and that there were no vaccines available, that people started realizing the serious nature of the situation we were in. And because identification is so important, we spent a huge investment in the TPCP doing correct taxonomy on different pests and pathogens, sometimes in advance of disease outbreaks, so that we are prepared. And here I will give 3 examples. The first one relates to pathogens on pine. Just last year alone, we described 4 new Lechinostecta species from Central America on pine, and some of these pines we are now using for plantation purposes. Currently, 8 of the 9 known Lechinostecta species are confined to this region, contained if you want to call it this, and we hope it remains this way despite increased movements and plant trade in and out of these native pine areas. Our second example is Calanectria, well-known pathogens of eucalyptus in plantations and nurseries. In just the last few years, 20 new species have been described by 3 of our PhD students on the bottom right-hand side. These 2 examples emphasize the amount of unknown biodiversity there is out there and the potential pathogen threats that are beyond our borders. But it goes further than this. We don't only look at species level, but also at a subspecies cryptic species level. And an insect example of this is Leptocybe invasive, that you all know very well, a gall-forming wasp of numerous eucalyptus species. For years we thought we were only working with one species until we used molecular methods to characterize the populations. We now know that there are 2 different lineages that are moving around the world in the invasive area, and that today we have both of them in South Africa. This knowledge has critical impact on, for example, breeding for resistance, as we know we have resistant clones for lineage A, but we're not so sure if the same is now true for lineage B, or whether the biocontrol we have been using for lineage A is as effective for lineage B. This and the research considering the interactions of these lineages with other hymenopterans is also work that is being done in the TPCP. Moving on to the third research themes, we might want to look at the origin of the species or the source of the epidemic, or track the global distribution and spread. This map on the left-hand side you have seen. But we might also want to look at the genetic diversity of individuals and populations, and we can use a genomics approach for this. And in this bottom picture on the right-hand side, you will see that over 5,000 individuals have now been sequenced of the novel virus, and the colors represented here are similar to the colors on the left-hand side in the map showing the global distribution. But what we can see from this picture is that there already is a lot of variation in the population that has accumulated in a very short period of time, and this can be due to mutation or drift. But it is very important that all of this variation needs to be considered if you are designing diagnostics or vaccines for it to be effective. And linking onto these themes and similar research we have done in the TPCP, I'd like to highlight one insect and one pathogen example. My insect example is Gonipterus. It is such a cute insect, but unfortunately, it really does some serious damage. The story here is that the beetle has been present in South Africa for now over 100 years, but the efficacy of the biocontrol agent Anaphes nitans is not optimal, and it seems to be breaking down as we are experiencing continual problems with the beetle in plantations. But now that we have sorted the taxonomy and we know which species occur where, we also know where the origin of both the beetle and the wasp is, and now we can go back to these areas to find more accurate biocontrol. In its native range in Australia, the distribution of the beetle and the biocontrol match quite nicely over a broad climatic range, as indicated by the circles. But the collection released into South Africa, however, is only from one site in Australia, there with this little green triangle, that has a Mediterranean climate, and it doesn't match the sites in Zululand in South Africa where we have the problems and which is more subtropical. So research at the moment is focused on gaining a better understanding of the diversity of the parasitoid and Gonipterus in South Africa and in its native range, and using microsatellite markers we are developing to gain this understanding in order to better match the biocontrol agent to the host here in South Africa. So understanding this environmental interaction as well as the biological interaction is very critical in this case. A fungal example is Teratosphaeria that causes both a leaf and stem disease of eucalyptus. Several of them are important forestry pathogens, and Teratosphaeria destructans is possibly emerging as one of the most serious threats to plantation forestry globally, and you can see it on the top right-hand corner of the picture here. For this group of pathogens, we are interested in unraveling the mating strategy of these fungi, and we have done this by using a genomics approach. And this is important because if sex is occurring in the field, it influences the ability of the pathogen to adapt to different environmental conditions. So the mating strategy has been determined for the stem canker pathogens and for the leaf pathogens, and both showing that they are heterothallic. This means that you need two individuals of opposite mating to get together or in contact with each other before mating or sex can take place. If we're looking at the population genetics of Teratosphaeria destructans using microsatellite markers that we have developed, we know that the population is clonal in South Africa and only has one mating type. The key message here is that there still exists an opportunity to plant tolerant clones because as it stands currently, the pathogen population is not able to change. My last research theme relates to biology and ecology and how this information can help inform us as to the invasive potential of pests and pathogens. Again, relating this back to the novel coronavirus, in terms of host jumps, this would be a bat to an intermediate host, such as the cats to humans or bats to camel to humans. In case of the novel coronavirus, we don't know what the intermediate host is, and this is an area for research. In terms of the life cycle, this would relate to how quickly the virus can replicate or make new viral particles, the viral load, for example, and how fast it could potentially spread. For the environment, we know that the disease is more prevalent in the winter and less so in the summer. And again, I have one fungal and one insect example to illustrate these themes. And I'll be starting off with myrtle or eucalyptus rust, really spectacular symptoms with bright yellow spores. But the problem with this pathogen is that it affects the new shoots, as you can see on the left-hand side, and the reproductive organs, as you can see on the right-hand side of the plants. And this has caused the near extinction of some of the Mutaceae species in Australia. Since its first incursion in 2013 in Gauteng, Ostracoxinea acidii has expanded its geographic range in South Africa, and this is over a wide range of climatic zones. It also infects several different native and non-native Mutaceae hosts, so it seems to be quite well adapted in South Africa. This invasion success could be attributed to its biology. In this case, some work done by Alistair, and welcome Alistair if you're joining us today, and the team has showed that the pathogen can complete its life cycle on a single host, and that sexual recombination is occurring in the field. So why is this important? Well, Ostracoxinea acidii is not a problem on eucalyptus in South Africa at the moment, but if recombination occurs, it does mean it has the potential to overcome this existing resistance over a period of time. But for now, it remains crucially important for us to continue monitoring to make sure that we do not have incursions of the more aggressive strain that affects eucalyptus coming into South Africa. With the last example looking at the theme of biology and ecology and how this might influence invasion success, the Sirex example works quite well. Here the TPCP group is looking at the driving factors that influence sex ratios in Sirex. This work was initiated after observing that the male-female ratios in the Western Cape, indicated in blue, changed over time, basically a 10-year period, and it changed back to what is normally found in the native ranges of the wasp. But that the populations in KZN and Mpumalanga in yellow, in the same period of time, remained skewed towards males, thus reducing the population growth rate in these areas. So for some reason, in the eastern part of the country, the females don't fertilize the eggs as often, and this seems to be environmentally influenced, that they have not really adapted to these regions. So the key message here is understanding the range of environmental factors that might influence the invasion ability of a species is important, especially for control. So looking ahead, our actions don't always have to be reactive, and that we can actually be proactive in the way we look for possible future threats to our industry, being one step ahead, the Red Queen analogy that Mark often refers to. And if I had to pick an example illustrating this, it would be the work that we are currently doing on pine needle diseases, and in particular, Dothrostroma needle blight. This is a disease that is gaining substantial international traction at the moment due to increased disease incidence and severity in many countries, particularly linked with climate change and increased planting of susceptible hosts. In the case of Dothrostroma, we actually have all the pieces of information I have just discussed with you in terms of those research themes. We know its identity, we know its distribution around the world, its interaction with the hosts, and we know how it is changing. And we can use this information now very practically to look ahead. And this is what we're doing. In South Africa, it is pretty well managed due to the fact that we don't plant the susceptible species. But this might be changing with the increased planting of Pinus techinomanii. But I'd like to draw your attention to the Central American strains. And these are very different to what we have in the rest of South Africa and the world, actually. And there are some Colombia strains that are quite close to these Central American strains. In Colombia, the disease is very severe on Pinus techinomanii, Urcarpa, and becoming more so on Pinus maximinoi. So the question we have now is if this very aggressive strain in Colombia poses a threat to the South African forestry industry. And so in a collaborative effort with Smurfit Kappa, York Timbers and the TPCP, we have done a pilot trial whereby we have planted some of our pure and hybrid species in highly affected areas in Colombia. Tentative results indicate that all species do become infected, but that Urcarpa and the Patula Tech low elevation hybrid is quite susceptible. So here we can be proactive in the species choices we make in the future if needed. To wrap up, I'd like to circle back to this slide. As much as the world has joined forces in an attempt to flatten the curve of the COVID-19 pandemic, which is a global problem, I hope I have shown you throughout this talk that so too is the burden of tree health, a global problem. And with that, FABI and the TPCP have very strong international research networks where we are working collaboratively together with the unified goal of keeping trees healthy. Thank you.